we started this about a month ago, and this grows out of the journal club that we've been holding at Johns Hopkins for the last 4 years, just on one of the Wednesdays every month. So this has just been a monthly club until now, and we usually get together and have some pizza or sandwiches and talk about EVs. But since many of us are in shutdown now, we decided to turn this into a virtual journal club, and we've enjoyed it so far, except for an incident of Zoom bombing. So hopefully we've controlled all of that now, because we really want to pay attention to our guest today. And I would like to introduce to you Angela Zivkovic. She's at the University of California, Davis, where she's an assistant professor in the Department of Nutrition. And I met Angela a few years ago because I came across some things that she had written about lipoproteins, especially HDL, and the different kinds of HDL that there are. So I learned a lot from her when I first talked with her about HDL, and I think that many of you are looking forward to that today too. So Angela has numerous interests, obviously nutrition, because that's her department, but she's also interested in neurodegenerative diseases, so Alzheimer's disease and some apolipoproteins. And she's also a member of the NIH's Extracellular RNA Communication Consortium, Stage 2, where we're working on separating the various carriers of extracellular RNAs. Angela has been very generous to us today and giving us her time, and I know she put a lot of time into getting this presentation together as well. So Angela, welcome, and we're looking forward to your comments and your knowledge on the extracellular vesicle cousins that we know as lipoproteins.
Thank you so much for that kind introduction and for adopting me into the greater EV community. I really have enjoyed seeing how this community works together. I'm very impressed with how organized and connected you guys are. So as Ken mentioned today, I will give you guys a bit of a background about lipoproteins in general. Their biology is fascinating, and some of the things that we still don't know. There are some things still that we haven't learned yet about these very well-researched particles. So I will give you guys a little bit of a story today. First of all, the reason that we know so much about lipoproteins is because actually cardiovascular disease is the #1 killer in the United States. It has been for a very long time, and it remains so still to this day, despite all the very successful treatments that we have come up with. And this is in large part because we know that the cholesterol-carrying low-density lipoprotein particles, or LDL particles, are causally involved in the process of atherosclerosis and the formation of arterial plaque. So they basically are involved here in the initiation of lesions, where when they enter the subendothelial space and are oxidized and modified, they're taken up by macrophages, which then transform into foam cells and then kind of generate that whole domino effect that results in the buildup of arterial plaque. Oops. Okay. So the story of LDL particles is actually quite easy to follow, and it has been a very satisfying story. It has been, peppered with Nobel Prizes along the way. And it started with the basic discovery that high total levels of cholesterol in the blood are associated with increased cardiovascular disease risk, a very simple observation which led to all the rest that followed. We then learned that cholesterol was actually carried by multiple different particle subtypes, and when we started separating them out, and whole dissertations were spent on calculating Svedberg flotation rates and density range cutoffs. And we figured out that the high cholesterol problem was really associated with the LDL particle. The higher the content of cholesterol carried in LDL particles, the higher the cardiovascular disease risk. We then discovered that this class of drugs called statins, the cholesterol synthesis inhibitors, did a fantastic job of regulating levels of plasma LDL cholesterol, and that once you reduce the LDL cholesterol levels, you also decrease cardiovascular mortality. we discovered that this was a pretty simple, in essence, now in retrospect, metabolism pathway where you have VLDL particles that are synthesized by the liver. The goal of these particles is quite simply to deliver lipid to the peripheral tissues. They come out as VLDL particles. They're very low in density because they're so full of triglyceride when they're first secreted. And as they're metabolized, as they go through their metabolic pathway, lipoprotein lipase cleaves the triglyceride that's inside there. All the fatty acid go to the adipose and skeletal and other peripheral tissues that need that fat for fuel. And as these particles get smaller and smaller, they get denser and denser. And they end up as LDL particles, which basically are just little balls of cholesterol. And the longer they circulate around, the less they're being cleared out of the circulation, the smaller and denser they become. And that's basically where the problem really comes in. They sort of become these old beat up particles. We now have newer diagnostics that actually look at LDL particle count and size, and they're even more successful at delineating risk for cardiovascular disease than measurement of LDL cholesterol ever was. Mendelian randomization studies have shown the clear causal link between LDL and cardiovascular disease. And there's just been, success after success in this field. There's new drugs out now that, for example, PCSK9 inhibitors. There's also the statins, which still work gangbusters. And we know that the lower the LDL, the better. And it's just been quite a success story. So we do know quite a lot about lipoprotein biology in general. Unlike EVs, just to take a quick pause, they are not phospholipid bilayers with a hydrophilic core. Instead, they're phospholipid monolayers with a hydrophobic core, packed typically with cholesterol, esterified cholesterol in the core, and obviously also triglyceride in the larger triglyceride-rich particles. The lipoproteins range in density from about 1 to 1.21 grams per milliliter and range in size from as small as 7 nanometers with the smallest HDL particles all the way up to as large as a micron or even bigger with the chylomicrons. Chylomicrons are the intestinal derived particles which are involved in absorbing all the dietary lipid. And then, as I showed you already, the VLDL are converted to LDL in the process of dropping off their lipid content. And then, of course, the HDL particles are the particles that are known the most for being the ones that take all the excess cholesterol back to the liver for excretion. So here's all the many things that we know in terms of lipoprotein biology. We understand the protein and lipid content and composition of all the different particles. We know their kinetics and their turnover. There's been many stable isotope studies done. We know the concentrations in plasma under a number of different physiological conditions and disease states. We have cutoffs. We know that when your LDL cholesterol levels are below a certain number, here's how much risk you have. When your HDL cholesterol levels are below a certain number, what the risk is in males and females and premenopausal and postmenopausal women. We know how they're metabolized, the receptors that are involved, the key apoproteins that are involved, how some of these apoproteins can become modified, oxidized, glycated, and other things that can happen to them along the way, and how that affects their functionality and their recognition by their receptors. We know that there's one copy of ApoB100 on each VLDL, IDL, or LDL particle, which makes it really nice and easy to count their numbers. We also know that there's one copy of ApoB48 on intestinally derived chylomicrons. And we also really are lucky we have all the antibodies and the X-ray crystallography of all the proteins that associate with these particles. So there really is quite a wealth of information available. So, well, this is really boring. Is there anything that's still left to be discovered? Well, it turns out there is a lot still left to be discovered, despite the fact that we already have this really solid foundation. The HDL particles are really where most of the discovery still remains. We do know that these are the particles that efflux excess cholesterol from the periphery, bringing it back to the liver for excretion. So simplistically here in this diagram, the liver is secreting the VLDL for delivery to the tissues of lipid, and then HDL is what's bringing the excess cholesterol back to the liver. We also have a really large literature base showing that high levels of HDL cholesterol are negatively associated with a number of chronic disease conditions. And that includes cardiovascular disease, of course, but it also includes Alzheimer's disease, a number of autoimmune conditions. And this actually holds true across populations. So this is not something that is limited to the United States. It really is found across populations. We also know that high HDL increases survival in conditions like sepsis. So if you are ever so unlucky to be in a septic condition, if you have high HDL, you're very likely to survive. And if you have low HDL, you're going to have a tough go. They've also found that centenarians have really high levels of HDL, so they definitely are predictive of longevity as well. One of the breakthroughs that happened was that we found through the many genome-wide association studies that were done in this field that the loss of function of an enzyme called cholesterol ester transfer protein, or CETP, is associated with increased HDL cholesterol levels. And so what we did, of course, was what we usually do, which was that we found an inhibitor of this enzyme to see if it would work and if it would increase cholesterol levels, HDL cholesterol levels. So Big Pharma invested quite heavily into the development of these CETP inhibitors. And unfortunately, it was a complete failure. So it turned out that the CETP inhibitors did raise HDL cholesterol levels quite significantly. They even increased their HDL cholesterol efflux capacity, but this didn't really lead to lower risk. And unfortunately, what happened was that the first generation of these drugs actually killed people in phase 3 trials, which of course has the tendency to pour a bucket of cold water over any kind of research endeavor. So unfortunately, pharma kind of abandoned this area, and you say the word HDL and they still go running in the opposite direction because of how badly this turned out for them. Unfortunately, it really did slow down research in this area. However, it did open up the research in terms of we now are going after really trying to understand what the different types of HDL particles are, not just raising the total amount of them in the bloodstream. To make things even more exciting, there is a U-shaped curve. So not only is it, the case that we can't just measure small or large HDL particles and know whether more or less of one or the other is good for you. It turns out that even the amounts of HDL particles are not exactly clear as to what is the optimal thing. So we know that too low of an HDL cholesterol level and too high of an HDL cholesterol level are both associated with negative outcomes. So there's this sweet spot in the middle, and we're really just trying to figure out how to actually improve upon the situation and use our ability to investigate HDL particles to improve them in individuals and to improve health outcomes. So why are they so complicated? Well, HDL metabolism is not unidirectional like it is with the ApoB containing lipoproteins. The nice thing about the ApoB containing lipoproteins is that they start out as large VLDL particles and they are continuously remodeled into smaller and smaller particles until they're finally taken up. They have this one route by which they're metabolized. HDL particles, on the other hand, can be recycled and remodeled a number of different times as they circulate through the bloodstream. They even spend some time in the lymph. Little known fact and little known appreciated fact is that they, in fact, when they cross into the subendothelial space from the bloodstream, they don't go right back into the bloodstream through the endothelium. They end up percolating through the lymph before they go back into the circulation. So they're continuously remodeled and they interact with all of the cell types. They interact with the bloodstream components. They interact with the lymph components. They interact with the extracellular space components. They're even internalized and resecreted by most cells that have been looked at so far. So they really are quite complicated little things. You never know whether when you're looking at a small particle, it's a brand new particle that just became synthesized and is a nascent particle, or whether it's one that was big and then was metabolized and became small again. Proteins and lipids are exchanged all along the way. They don't just have a single ApoA1 copy per particle, like the convenient ApoB particles do. There could be 1, 2, 3, 4, or 5, depending on the size of the particle. And there's even certain particles that are containing ApoE or ApoA2, but not ApoA1. So you can't even just say, well, here's the putative receptor, or excuse me, protein that I will isolate them with, and then I'll know I'll have HDL particles. You might be missing out on a whole set of particles that don't happen to have ApoA1 on them. Some of the recent studies have even found that when they separated according to protein content, when they used immunoprecipitation and ELISA-based methods to using antibody capture, that there may be as many as 16 different subclasses with distinct protein cargo and functions. So we think that these little particles are actually platforms that have on them different subsets of proteins that work together to perform very specific functions. They're multifunctional particles, and what we know is that they have a range of different functions, some of which are mediated by their ability to efflux cholesterol out of the cell, and particularly out of the plasma membrane, where they can, for example, deplete or disassociate certain proteins, for example, B cell receptors and T cell receptors and the TLRs, dimerize by sitting together on a lipid raft. And so when HDL particles come along and take cholesterol out of the membrane, they disperse these lipid rafts, and these different signaling proteins that normally like to sit on lipid rafts and interact through lipid rafts are now dispersed and no longer joined together. And so HDL can have a lot of anti-inflammatory and immunomodulatory effects simply through their ability to efflux cholesterol out of the membrane. But they also have additional effects through some of the other proteins that they carry. For example, they have antioxidant functions through some of their enzymatic proteins, the PON1 protein, for example. They affect endothelial function. They have anti-apoptotic effects, antithrombotic effects. And so they do perform a lot of different functions, and we're really still just discovering all the things that they do. What's fascinating about them is that we know that they interact with all peripheral cells and blood cells, and they really have a crucial function in determining the function of those cells. HDL can even become dysfunctional. We know this really well, that both chronic inflammation and acute inflammation can cause HDL particles to go from these happy little particles that efflux cholesterol out of cells and inhibit oxidation to particles that instead are remodeled into pro-atherogenic particles that have reduced ability to perform all those beneficial functions, and in fact can have a pro-inflammatory effect. And unfortunately, this is not something that we have seen to be correlated with things that are easy to measure, like HDL cholesterol levels or even the particle size. So we know that HDL composition and structure dictate function, but it really depends how you isolate the HDL particles to get the answer that you end up finding. So for example, the HDL proteome may consist of as few as 12 proteins, or it may be over 100 proteins, depending on how you isolated those HDL to begin with. I'm just kind of showing a couple of different ways of isolating HDL particles that have been published in the recent years. This one here involves a sequential immune-based or antibody-based capture. This one here involves size exclusion chromatography followed by a resin binding, which basically pulls out all the things that are bound to lipid. There's another one that uses density gradient ultracentrifugation followed by size exclusion. This one here certainly gives you the purest HDL fractions. It involves ultracentrifugation followed by size exclusion chromatography followed by antibody capture of ApoA1. And it definitely gives you the purest fractions, but it may be missing some of the larger HDL particles that might be co-eluting with LDL, and some of the smaller HDL particles that might be co-eluting with albumin. And it might also be missing some of those HDL particles that I mentioned previously, which are the ApoE only particles and the ApoA2 only particles. So there's, disadvantages and advantages to all the different methods, and what everyone is starting to realize is that this combinatorial effect is really what's going to be needed, because any one of these methods alone doesn't seem to be adequate for adequately purifying these protein particles from the blood. So this is just another way of showing how, depending on the HDL isolation technique, you might be grabbing a different subset of the total HDL pool from any given serum or plasma sample. For example, here, the isolation by antibody capture of ApoA1 versus ApoA2 containing lipoproteins breaks the particles down in this way, versus if you separate them by ultracentrifugation, what will usually come out in the HDL2 particles is captured here in this green triangle, versus the HDL3, which are the smaller particles, will come out here. So you can see how, depending on exactly what method is used, you're going to be grabbing a different subset of the total pool of HDL that are present in the serum. And it's very conflicting out there, what you can find depending on what the HDL isolation method was. This is just another way of looking at this. Of course, the particles do carry charge, and so gel electrophoresis has been used to separate them by charge. And they can also be separated by size and by density. You can accomplish this separation of all of these different subclasses of HDL by ultracentrifugation alone, but very prolonged ultracentrifugation times are required for that. And we know that there's loss of certain proteins and modification of the particles with that type of extensive ultracentrifugation. So we're basically just starting to scratch at the surface of how complicated these little particles are, as much as we know about them. We do know that they have a variety of different and sometimes opposing functions that, of course, depends on what proteins are present on the particles. We know that they're continuously remodeled, that they have a complex proteome, that there's subclasses, that they have a variety of lipids, and that, of course, they carry RNA particles. And I will come back to that. First, I just want to mention real quick a really interesting angle to this, which is that it turns out that these little particles are highly glycosylated. Ignore those textbook pictures that show HDL particles as this naked thing. They are completely covered in glycans. They're covered in glycans that are N-glycans, O-glycans, these little tiny guys, as well as sialylated glycolipids. So it looks like over 70% of the proteins in plasma, as well as about 70% of what's found on HDL particles is in fact glycosylated. ApoA1 is not, interestingly, but just about every other protein that associates with HDL particles is glycosylated. Glycosylation, of course, drives many critical functions. I'm not going to spend too much time on this, but I just want to give a really quick background to make sure we all know what these wonderful little colorful things are that we're looking at. Basically, the N-glycans are represented here and the O-glycans are represented here. They are two different forms of post-translational modification. N-glycosylation typically involves larger, more complex structures, and the sites of attachment are attached to a consensus sequence, amino acid sequence, so they're kind of easier to find. O-glycans, on the other hand, are very tricky because really any serine or threonine residue in a protein could potentially contain an O-glycan on it. There's multiple sites of attachment of these different glycans on many proteins. Some proteins are only O-glycosylated and only have one site. Some have both N and O glycans and have multiple sites. For example, ApoB100 has 11 glycosylation sites, some of which are N glycan sites and some of which are O glycan sites. And there's many possible glycan attachments that can happen at each site. So actually measuring quantitatively how much of each type of glycan is attached at each site is something that is possible now and that we do as part of our group. So just as a reminder, glycans, of course, are important in mediating binding recognition between cells or between cells and viruses. I thought this was really apropos, so I chose this figure here. They're involved in adhesion, endocytosis, protein folding. fascinating, they even act as a clock. So there is an asialoglycoprotein receptor on the liver, which basically recognizes when a protein is old because it has lost its sialic acids and it's just cleared by the liver. It means it's old and it needs to get out of there. So the glycan attachments on proteins are very fascinating and very informative and important in driving a lot of these really critical functions. It turns out glycosylation is associated with HDL function. So one of the first studies that we did was we looked at a couple of different patient groups. We looked at healthy individuals with metabolic syndrome. So they had multiple traits of metabolic syndrome, low HDL, high waist circumference, high blood pressure. And then we also had sort of as like the end of that path, if you will, diabetic patients with chronic kidney disease who were on hemodialysis. So these were really quite sick individuals, right? We isolated HDL from all three of these groups and we did LC-MS glycoprofiling. We only had 3 proteins that we started with. It takes quite a lot of effort to develop methods of site-specific glycosylation profiling. You have to do it protein by protein, and it's a very laborious task. So we started with 3. And then we also measured HDL anti-inflammatory capacity. Basically, we took macrophages, we stimulated with them with LPS, and then we incubated HDL to see how much the HDL isolated from these different groups would inhibit the IL-6 secretion by the stimulated macrophages. And we couldn't believe the results because I thought this was going to be very simple. I was going to do a graph of healthy versus metabolic syndrome versus the hemodialysis patients. And clearly the hemodialysis patients would have the least anti-inflammatory HDL. The metabolic syndrome patients would be somewhere in between, and the healthy controls would have the most anti-inflammatory HDL. And it was not the case at all. We actually did not find that the anti-inflammatory with any of the typical clinical parameters that we measure, like BMI or even total cholesterol, HDL cholesterol levels, any of the CRP or anything like that. What we did find was that HDL glycoprofiles were highly diagnostic of whether someone's HDL particle was going to be pro or anti-inflammatory. And we found that there were multiple modifications on these different proteins that were found to be enriched in HDL particles that were anti-inflammatory versus HDL particles that were pro-inflammatory. We also found that HDL glycosylation is associated with cholesterol efflux capacity. So this time we're basically using another in vitro assay, which we take macrophages, we load them with fluorescently labeled cholesterol. We then incubate HDL particles, and we just simply measure how much of that fluorescent cholesterol is efflux to the HDL particle, and then just calculate a percent efflux capacity. This here is just showing in a little bit more detail what the glycoprofiling looks like. So it's a targeted LC-MS method, and we're using multiple reaction monitoring to simultaneously quantify not only the amount of each protein, but also all the different glycosylations at each site. This is work that is done in Carlito Labriglia's laboratory here at UC Davis. He's in the Department of Chemistry, and he is a world-renowned Glycan analytical chemist. And it has been my absolute privilege and good luck to have had the chance to work with Carlito over these years. His methods are really quite amazing. So in this case, we monitored five proteins because we were slowly adding proteins to that method. And now we include ApoE, which of course is really interesting and fascinating because of how important that protein is in determining risk for diseases like cardiovascular disease, and in particular, Alzheimer's disease. We discovered that ApoE doesn't just have 1, 2, or 3 glycosylation sites like it was thought. It turns out it has 6 glycosylation sites, actually, no, excuse me, 7. And that one of these sites, which wasn't even known before, is the one that is the most complex and has the most possible different types of glycan attachments on it. So we did find that out of the 80-something glycopeptides that we monitored in this study, a good proportion of them, so 25% of them, were actually correlated with HDL cholesterol efflux capacity. And some of these relationships were actually quite strong. So these correlations really held up. And it looks like HDL glycosylation might be influencing efflux capacity as well. And then finally, because as Ken mentioned, I'm in the nutrition department and we do nutrition intervention studies, we did a study where we fed people, I kid you not, a fast food diet, which involved eating a Pop-Tart for breakfast and Carl's Jr. burger and fries for lunch and dinner, versus a nice, healthy Mediterranean diet, which included all these wonderful fruits and vegetables and whole grains and things like this. And we found that after just 4 days of feeding this diet, we saw pretty big changes in the glycosylation, not across the board, but only of very specific proteins. And in fact, it was really only the ApoC3 protein whose glycosylation was altered. If you follow the HDL or, excuse me, the lipoprotein literature at all, you'll know that ApoC3 is kind of a hot topic, and it seems so to see that diet can have glycosylation like this in just 4 days is really quite astounding. I just wanted to show really quick that we also do other omic analyses in the lab. We work with the West Coast Metabolomics Center to do lipidomics, and we found that the HDL lipidome was really completely remodeled within 4 days. So basically what you eat in terms of the lipid content is going to almost completely remodel the lipid composition of your HDL particles quite quickly. We see this happen even in the postprandial state, but certainly within 4 days, we see that the fatty acid composition, the phospholipid composition is going to be reflective of the diet that you're eating. Okay, so that's all good and fine, but how does this have anything to do with extracellular vesicles? Well, I have two ways that I decided to try to illustrate this to you guys. First is that, like EVs, lipoproteins are also extracellular RNA carriers, so we should care about them simply because of that. And we should also care about lipoproteins if we're EV people, because when isolating EVs from plasma or serum, lipoproteins are a major source of contamination. So first of all, I'm showing here just the xRNA atlas that was one of the products of ERCC1. And of course, lipoproteins were included in that analysis here. And the micro content of lipoproteins was analyzed and was found to be unique and different from EVs and from the other carrier types of extracellular RNA. So some of the interesting questions here are, what might be some of the shared biology of EVs and lipoproteins? Do they have common endocytic pathways? Do they actually exchange materials between each other intracellularly and maybe in plasma, the way that the lipoprotein particles share materials with each other and interact with each other? Can we apply the 50+ years of lipoprotein research to inform some EV research? So for example, we know quite a lot about how diet and lifestyle affect lipoprotein composition and function. Can we use some of those same, sort of ideas and concepts and study designs to look at whether some of these things also affect EV composition? And then there's also a lot of tools that have been used in the lipoprotein field that would be very useful for EV research as well. For example, we use lipidomics, proteomics, glycomics, to characterize the cargo of lipoproteins, and similarly we could do that with extracellular vesicles as well, to add to the already very cool picture that we have of what kind of RNA they carry. Also, there's been some really elegant double labeling experiments done with lipoproteins where we've tracked particles to see exactly what their turnover is and what their metabolism is, where they're secreted and where they're taken up. So there's a lot to be learned from the lipoprotein field, and it's, like I said, a very long and very full research field. And then, of course, importantly, it's really key to recognize that, first of all, lipoproteins outnumber extracellular vesicles in plasma by several orders of magnitude. There are a lot of lipoprotein particles in the plasma compared to how many extracellular vesicles there are. And you have two problems. HDL overlap with EVs in density. So if you isolate EVs by ultracentrifugation alone, there will be tons of HDL particles in there contributing to the RNA content that you're probably analyzing. Also, VLDL and their remnants overlap EVs in size. So if you use size extraction alone, your EV fractions will be contaminated by a ton of the ApoB containing lipoproteins. And so this is, I thought, just a nice illustration to show, that the EVs here fall into the density range of HDL particles, and they also fall into the size range of some of the remnant particles as well as VLDL particles. So what method we've come up with in the lab for isolating HDL particles is basically a sequential method that uses sequential flotation, ultracentrifugation, followed by size exclusion chromatography. So we separate the particles first by density and then by size. And we do this in a unique way. We opted for sequential flotation because it really minimizes ultracentrifugation times. So in total, our particles are only spending about 4 hours in the ultracentrifuge compared to a minimum of 6, 8. Some of the older ultracentrifugation techniques with the older rotors and the big bucket and the big tubes, you're basically spinning these things for 3 days to get at the HDL particles because they're the densest ones and the hardest to get out. So what we do the VLDL, the really sort of goes out of there first. And then we do a second spin to basically precipitate out all the plasma proteins and the albumin as much as possible. We're then left with this fraction here that has the density range that encompasses all of the HDL, but it includes the LDL and the EVs, and we put that on our FPLC. And we basically are able to isolate the LDL and the HDL particles, and we get a really nice concentrated fraction of HDL that we can break up into 4 separate aliquots that then can be, subjected to for example, 3 different compositional measurements and a functional measurement, all done on the same technical aliquot of HDL. And we purified, or I should say, we verified the purity of our HDL particles from this extraction. We did gel electrophoresis just to confirm that we don't have a bunch of junk in there. We did shotgun proteomics, and we also analyzed these fractions by electron microscopy. And we do also see some of a really nice purification of those HDL. And then as I started working with Ken, we were curious just because it would be so convenient if this method that already isolates the HDL particles, we could also capture the exosomes and the EVs and basically just not have to throw them away from a potentially very precious clinical sample. So we wanted to see whether they would be eluting right here in front of our LDL particles, which is where they should be, because the density range of EVs, of course, is 1.1 to 1.9, and the density range that we actually injected onto the UC is 1.006 to 1.21, so they should be right in there. So we took these fractions and we sent them to Ken and Emily Malik and others in Ken's lab. were able to analyze these particles, and they found by electron microscopy that indeed we do have EVs in there. They have the putative size and sort of appearance of them, or at least what Ken tells me they're supposed to look like. They also did some spectrodite analysis and showed that there were low but definitely measurable counts of EV-sized particles in there. And then finally, they used ExoView and were able to detect the presence of antibody-specific particles in there. So we are pretty convinced that we are in fact grabbing some EVs. And so it may not be the ideal way to isolate EVs, but we certainly can make sure we don't throw them away from a precious clinical sample when we're isolating our HDL particles. So we're very excited about that, and hopeful that might be useful, and that we can look at it further. So for example, right now we have a sample set of 220 Alzheimer's disease patients who are all clinically defined, but also biopsy confirmed, or not biopsy, confirmed from the biorepository that they are in fact Alzheimer's patients, ApoE genotyped. This is a really precious resource that we were able to get our hands on to do HDL analysis, and we are able to keep those EV fractions, and we're hopeful that we'll be able to analyze them and find something interesting there. So basically, long and solid track record of research history that could be very informative and that could be applied to EV research as well. And we're very excited to, apply some of the techniques from plasma or serum. Really need to very carefully account for this. And then, the co-development of isolation and characterization approaches for lipoproteins and EVs, I think will really benefit both fields. I'm very excited to be involved with the EV community, and I feel like I've already had my eyes open to so many new techniques that I didn't know existed before. And in fact, as a quick sneak peek, Dr. Wyatt Breland, who is a collaborator on our ERCC2 project, who is at NIST, is using asymmetric flow, field flow fractionation, or AF4, to separate EVs and lipoproteins because it's such a gentle separation technique that results in minimal particle degradation and loss. And when it's paired with light scattering detectors, you can actually also analyze these particles at the same time. You can analyze their size and count while isolating them from each other. So it's a really powerful new technique, and I am super excited to have a chance to work with Wyatt to look at these particles with this new technology. There is a paper that was published recently by a group out of the CDC where they looked at, in fine-grained detail, in 1 to 2 nanometer increments of HDL and LDL particles. And then they applied some really nice advanced LC-MS technology to look at the composition of those tiny little sub-fractions of LDL and HDL particles. And this, this is actually an article that we're spending, several weeks in our laboratory discussing, because it is so full of information that is really mind-blowing. For example, there's this whole type of particle here, the ApoE containing HDL particle, that's eluting, we now know, in this very specific size range that's been largely ignored in the HDL field because it just happens to be kind of hard to grab it. It tends to overlap with LDL particles in size and density, and it's a little bit bigger than the typical HDL particle. But it's actually really fascinating that we're going to have a chance to now look at some of these subclasses that we've never had a chance to look at before. So with that, I wanted to make sure and acknowledge all of the people in my lab, of course, as well as all of the collaborators that I work with and some of the granting institutions that have contributed to all of this research. And then I would be very happy to answer any questions.
Wow. Well, thanks very much, Angela. I have a lot of questions myself, but I think we've also gotten a lot of questions in the chat room. So I'm going to try to unmute some people and let us have a good conversation here. So I think I will start with Erez Aitan and I'm sorry, no, sorry, one second here. It was Roger Alexander. Erez, you're next. Roger Alexander and Massimiliano Rušička had some questions about the CETP inhibitor trial. So I'm going to try to unmute some people here. Maybe some of my co-hosts can help me with that. So it looks like Roger, you're unmuted. Do you want to go ahead with your question?
Yeah, so I was just curious if they know the specific mechanism that caused deaths in the phase 3. I guess after I asked the question, it was something about HDL concentration being too high, but like what happened?
Well, you know, they're still trying to tease that apart. The next generation CETP inhibitors were actually, they were able to avoid that off-target effect. It looked like it had something to do with effects on blood pressure. And so actually the mortality increase that happened with the first generation CETP inhibitors was related to infectious disease and non-cardiovascular mortality. So it was just an unfortunate off-target effect of the drugs that they were able to fix, but it just, you know, once it happens in a phase 3 trial, it really is very debilitating for that whole kind of line of research.
And Massimiliano, I think that I was having problems unmuting you, so I apologize for that. I'm not sure what's happening, but I did want to just note that you had mentioned that there is a new study going on. Wait, is this working now? Massimiliano, are you there?
Yeah.
Okay, great. All right. Sorry, I couldn't unmute you, but I'm glad you are unmuted.
So thank you for your talk. Just to point out that there is ongoing the DAL-gene study with the dalcetrapib that involved patients with the specific SNPs on not on the CATP but on another gene, and maybe there will be good results. But just my curiosity is if in your studies you have seen, or do you think that the activity of some pharmacological intervention inhibiting CATP can change concentration of extracellular vesicles? Just that. It's just a curiosity. Is a pharmacological intervention can change the amount of extracellular vesicles released in circulation? Thank you.
I mean, I think that's such a fascinating question. You know, my understanding of EV biology is really limited since I'm so new to the field and I'm just kind of learning. But I think that probably cholesterol levels might play a role, generally speaking, because I know that some of the endocytosis and exocytosis pathways depend on lipid rafts, and the cholesterol content of the membrane therefore is probably pretty important. But I don't know if there's any studies that have looked at levels of EVs in the context of changing levels of blood cholesterol or anything like that.
It was just a curiosity because you introduced the concept of CATP, just that. Okay, thank you very much.
Yes, of course.
Great, thanks for that question and comments. Erez, you're next. You had asked about the sweet spot of HDL, and then also about the comparison of Angela's techniques with commercial HDL isolation. Do you want to ask those questions?
Sure. So I was very curious from a biomarker perspective, if when you talk about a sweet spot of benefit to damage, what is the variability between individuals? Later, I also wanted to ask the same on the glycolysis of the HDL. You said that you find differences in the glycosylation with function, and I was very curious about the variability between individuals. And then the second question, we used commercial HDL from Sigma, for example. If you have any insight on how they make their HDL and how pure it is.
Yeah, so I'm not 100% sure how they isolate HDL. I have a suspicion that it's probably basically just ultracentrifugation. Whenever I've seen papers that use purchased HDL particles in their analyses, they always find ApoB contamination and albumin contamination. So to me, it looks like it's probably a basic ultracentrifugation isolation. I'm not 100% sure though. And then in terms of the variability, yes, there's huge amounts of variability. So, you know, here in this kind of idealized scan from our FPLC, we're showing this one, you know, curve that this is what it looks like. Here's how much LDL and here's how much HDL and here's what the width of the peak and exactly where it elutes. This is very highly variable, but between individuals. Some people actually have this kind of double peak here in the HDL because they have so much of the large HDL particles that they actually form their own kind of separate peak that's not perfectly resolved, but it's certainly a double peak. Some people don't have any large HDL particles at all, and they only have the small ones, and you can actually see a shift in that peak over to the right, and you can see that there's more sort of coelution with albumin, for example. Other people have, you know, hyperlipidemia, and you actually really see kind of a buildup of stuff here on the left side or the large side of the LDL particle curve. You can see that they have a ton of IDL that flowed into the LDL fraction and that expand that or broaden that peak. And we do also see a great deal of variability in terms of the glycosylation in individuals as well. We are starting to look at what that looks like in Alzheimer's disease patients, and we're definitely seeing a lot of variability there.
Great. Okay, so we next had a question from Gilman. I'm wondering if that might be my colleague Bob Gilman here at Hopkins, who asks, what is the effect of aging on...
Yeah, it's me.
Hi, Bob.
Just a question. Clinically, what happens with age and gender on these things? Gender obviously doesn't change, but age does. And clinically, what we, with at least on a few of us, we see decreases over time with HDL. Yeah, can you explain that?
Yeah, absolutely. You know, so there's a couple of possible mechanisms by which HDL particles are reduced. So, you know, CETP plays a pretty prominent role there, where essentially in the context of high triglyceride, CETP is in fact going to be transferring triglyceride to HDL particles in exchange for cholesterol that is then going to be going from the HDL to the VLDL particle. And so what happens as part of that metabolism, this actually might be where it's even better illustrated, is that the HDL particles, now that they're enriched with triglyceride, are kind of mistaken for an ApoB particle, and in fact are acted upon by hepatic lipase. And then that lipase is cleaving the, you know, the triglycerides and making those particles smaller. Well, what happens is that now you've got all this excess, you know, sort of surface material that's coming from the shrinking particle. And so some of the ApoA1s fall off. And once they're lipid poor or lipid free, they're more easily cleared by the kidney.
All right, okay.
Oh,
Cecilia, you can ask your question.
okay, perfect, thank you. So, I mean, I'm interesting to hear your view on how good are we at separating the EVs and the lipoprotein particles currently, and if we use these like ApoA and ApoB as marker of contamination of the lipoprotein particles when we're looking at our vesicles. I mean, some people have suggested that ApoE is actually present in vesicles, and I think Edith Buse's group has also looked at like lipoprotein particle like covering the vesicles. So do you think that we can like truly separate them and determine contaminations?
Yeah, I'm very hopeful that these new technologies, for example, the AF4 technology that Wyatt Reeland will be talking about in a couple of weeks in this journal club, that these kind of techniques will be able to allow us to really separate these things from each other in a gentle enough way so that we can trust that we're actually seeing separation and not the formation of weird aggregates or complexes or particles that have broken up into little pieces and things like that. I think I'm not sure that it's clear that ApoE is a component of EVs or whether it's a contaminant from some of these other lipoprotein particles that probably are co-eluting and overlapping in size and or density.
Yeah, I mean, I really think this is the challenge to determine what is a true contamination and where have they just been aggregates, and how do we do that, separate them in a good way. I just had a question also about ApoB100. I heard it's really hard to do that in Western blots to detect that. Have you succeeded in that? I mean, do you have any experience on that?
It might be tricky.
Because it's quite a big protein, right?
It's a very big protein and it's highly glycosylated. So as I mentioned, it's got 11 glycosylation sites. It's loaded with glycans. And so it does, sort of behave a little bit differently. But yeah, there are good protocols for doing Western blots for ApoB100. Okay,
good.
Yeah.
Yeah, because from the EV point of view, working with plasma, I'm always interested in, determine the contamination about the really fascinating or the fascinating lipoproteins that you're working with, trying to eliminate the vesicles. So
yeah, it's so funny. I mean, the reason I actually got involved with EV was because I was like, oh, great. Now I've got this new contaminant I have to worry about. So your guys' EVs are my HDL contaminants, right? So it's good that we're getting together and
that's perfect.
Together.
Yeah, thank you for a really nice presentation.
Thank you.
Thanks, Cecilia. So I think I'm back. I hope people can hear me. I was going to ask Simon Schofer, I think you had a question about where else we might find EVs, I mean, not EVs, but rather lipoproteins other than the blood.
Oh yes, hello, exactly. We are working with the urine and other substances, the human body, liquids, and I was interested in if it's the concentration of the lipoproteins is everywhere very high.
So urine really shouldn't have any lipoproteins. In other words, if you're sort of a normal healthy person, there should really be negligible amounts, although I have seen that people can detect both lipids and ApoA1 in urine. And I never published this, but as a graduate student, I actually did measure ApoA1 content in IgA nephropathy patients, which is a glomerular disease, and they have really low HDL. So I was very curious if what was happening was that they were sort of clearing them through the urine. And we did see a higher level of ApoA1 in kidney disease patients, but it should be very low quantities in urine. There is a whole parallel universe of lipoprotein particles in the CNS, the CSF, the cerebrospinal fluid. And they're basically sort of like brain HDL particles, which have ApoE as their main protein rather than ApoA1. And they're slightly different in terms of size and composition, but they're essentially kind of like HDL particles, and they sort of operate in their own universe over there in the cerebrospinal fluid.
Oh, okay, thank you.
Mm-hmm.
Great. So I've just unmuted Ursula Sandow. You had a question about glycosylation, and I think that we had a couple of questions about glycosylation actually, but we're running out of time, so I think we're going to make this the last question. Ursula, do you want to ask?
Sure. It's kind of related again to that CSF question or the CSF comment. So I was just curious, do you know if ApoE or ApoE in general has different glycosylation patterns based on the subtypes E2, E3, E4, because they infer different risk with Alzheimer's disease?
We are very, very interested in that question. It turns out it is not trivial at all to look at that, because ApoE2 and ApoE3 and ApoE4 are only different from each other by 2 single point mutations. So it's very, very difficult to tease apart those isoforms, and even more difficult than to actually monitor their glycosylation. So it's going to take a pretty large study with large numbers of homozygous and heterozygous phenotypes or genotypes, so that we can look at that in more detail. And that's definitely a direction we're going in, but we don't have the answers yet. As I mentioned, it was really just last year, at the end of last year, that we just published the first paper showing that ApoE has 6 glycosylation sites. Up until now, people thought maybe it had 2, maybe 3. So the type of glycosylation that's found on ApoE, O-glycosylation, is way more difficult to study to begin with because of the chemistry of it, and because it can just be on any serine or threonine residue. So it's much more complicated to get a site-specific glycosylation profile for O-glycosylated proteins. And then ApoE has this really complicated isoform structure. So it's going to take some time, but we're going to get there, I think.
Great, thank you.
All right. Well, thanks everybody for your questions, for your interest, and for showing up to listen to this very educational talk. I'm sorry to those of you whose questions we didn't quite get to today. We'll try to address those in the near future, maybe directly with you, or we'll put this on the YouTube channel. So please subscribe to our YouTube channel. I'll send out a link for that. This recording will be put there hopefully very soon. And so thank you all again. And I'd like to give a special thanks to Angela for sharing your knowledge with us. And we look forward to seeing you all again very soon. So stay safe, everyone, and take care. Bye now.
Thanks, Ken. Thanks, everyone. Bye-bye.